Okay, if everyone is settled, Professor Alexander will start our program on a lab of one's own fighting bioterrorism, cholera, and COVID-19. We are all set to go, so it's yours. Thank you.
Thank you very much, Bob. I'm Professor Wallace, and I'm chairman of the International Law Institute, and I want to welcome you to this webinar. Rita Colwell is an old friend of ours, and I welcome her. Yona will introduce both her and her book, and after Rita has spoken, there will be two commentators, and again, Yona will introduce them. So, Yona?
Thank you very much. I would like to join you to welcome Professor Rita Caldwell and our commentators, as well as the audience. Very briefly, I would like to introduce Professor Caldwell, who is regarded as one of the leading scientists, not only in the US, but also internationally, in honor of her exceptional contributions to global health and security concerns. And as you mentioned, she published the highly acclaimed book that I will try to show right here. As you can see, a lab in one's own, one woman's personal journey through sexism in science, which was published by Simon and Schuster the past summer. I will just say a few words now in introducing her, because she really is very exceptional in Washington. And I wanted simply to mention a few of the highlights. And I would like to introduce also briefly our commentators, and let Professor Caldwell discuss a very timely and important book. So again, some of the highlights, of course, academically, we had the honor to work with her. She is a distinguished university professor at both the University of Maryland and Johns Hopkins University, the Bloomberg School of Public Health. And of course, she is well known around the world, as I mentioned, for as contributions, pioneering contributions, I might say, microbiology and some related fields. Of course, she has a very long list of awards, all the way from over 60 honorary doctorates from universities throughout the world. And she was also awarded by the Emperor of Japan, the King of Sweden, the Prime Minister of Singapore, and the US President. But I think what is really important for all of us to know, that a focus on the global infectious diseases, water, health, and so on. And she's very practical in terms of trying to develop some international network to address some of these issues around the world. And hopefully she can discuss it during the session. And I might also mention that she co-authored more than 16 books, and probably 700 or more scientific publications. As academics, we are very honored to have the opportunity to work with her over the years, particularly going back to 9/11 with the anthrax challenge and all that. But at any rate, now some of us are reading the book, as I mentioned, that is available, and many comments are quite interesting, mentioning that it is a profound book, it is a compelling book, it is fascinating. I can tell you from our personal experience at home, we decided to have a little family book club, and we're discussing some of these issues that my wife and I, daughter and even son, who is not here present. So I think this is going to be a very timely and challenging book, especially this particular time. Now, before, as I said, I'm going to invite her to speak. I want to mention that we have also two very distinguished commentators. One is Dr. Winton Cerf, who is the Vice President and Chief Internet Evangelist for Google. As I understand, he is going to be a little bit late, so I'm going to invite now our colleague, Dr. Norm Kahn, who is currently the president on Counter Biology, which is a consulting firm dealing with security issues, particularly focusing on weapons, mass destruction, and biological weapons. And he has a very distinguished career with the Central Intelligence Agency. I think over 30 years or so, including multiple assignments in intelligence and technology and so forth. And if he wishes, he can discuss some of these issues challenging the past many years. At any rate, he's an academic and contributing to our work. He has a degree in biology from City College in New York and a PhD in oceanography from the University of Rhode Island. So with that, I would invite Professor Caldwell to tell us about her book, anything else that she likes to discuss, and then we're going to invite our commentators.
Thank you very much. I appreciate your introduction, Yona. What I've chosen to do is to really focus on Chapter 7, the anthrax letters, because it's a story that has not been told. It took place mostly, well, entirely in SCIFs, and there was a press conference, and it has been written up by some other authors, but I think this particular story is quite unique. It describes how men and women worked together and chose not to have any specific formal notification. We met informally. And so it really starts because my connections to the US intelligence community, the 17 government intelligence organizations, notably the CIA, began in the early 1970s. I was an associate professor at Georgetown University, but I had been elected president of several international biological organizations. And these organizations had meetings in South Africa during the time of apartheid. Organizations also met in Czechoslovakia. We met before the fall of the Berlin Wall, and a lot of other interesting places like that. I did a lot of work in South America as well. I wrote a lot about my research on cholera, which I started really as a graduate student working on marine bacteria, made the discovery that cholera bacterium actually is a marine bacterium. It's found in the environment. But in any case, my work on cholera and all these travels got me thinking about something that would interest the CIA, namely microorganisms as a weapon. a weapon of mass destruction, biological weapons, biological incidences, and potential biological attacks. So in the late 1990s, that's 30 years ago, I got in touch with John Phillips, a PhD chemist and the chief technical officer of the CIA. He later became the chief scientist for the entire intelligence community. And I asked him if there was a committee that addressed bioterrorism. And he said, no, there wasn't any currently. And my question was, shouldn't there be one? He agreed. But months went by, and a lot of us were advocating a database, at least, of biopathogens, because at that time, it was still pretty rudimentary. This was pre-sequencing. And so we didn't think anything was happening. But then in January 2001, quite auspiciously actually, I got word from John Phillips, who it turns out was not only a good scientist, but an effective administrator, that he'd gotten funding from Congress to begin a major research and development program for countering chemical and biological terrorism. And he invited me to join the advisory board. So I accepted, but nothing happened for 9 months. This was the 1st of January and first weeks or so of January in 2001. Then, while I was in my office as director of the National Science Foundation, I had joined the National Science Foundation appointed by President Clinton in 1998. So at 8:45 a.m. on the morning of September 11, I was in my office at the National Science Foundation, ironically, with 2 staffers from the White House. We were discussing the upcoming NSF budget to go before Congress, and the budget that was coming out of the White House to go to Congress. And suddenly, one of my staffers broke into the room, knocked on the door and opened and came into the room and said a plane had just crashed into one of the World Trade Towers in New York. And I flipped on the TV that I had in my office, and we all watched as the second plane crashed into the World Trade Center. And an hour later, we learned that a third plane, hijacked plane, had crashed into the Pentagon, which is only about a 5-minute drive, really, from the NSF office at that time in Arlington, Virginia. And then a fourth plane was over Pennsylvania, racing toward the White House. It was surreal. It was surreal. Just as a side comment, we weren't able to make any contact with the White House until 11:30 that morning. And I needed to let my staff, 1,500 people, know what to do. And so I asked to get the PA system. It turned out the PA system was in the basement, and the keys were owned by the fire chief from Arlington, who was busy at the Pentagon. So my first act then was to break the door, to order my staff to break the door, so I could get in to announce to my staff that they could leave if they wanted to, but I didn't recommend it. In any case, 3 weeks later, on October 5th, a committee on bioterrorism had its first meeting at the CIA headquarters. The nation was now on high alert after the 9/11 attacks. And on October 5th, 6th, and 7th, we had a very tense informal workshop about chemical and biological terrorism. And then a press release shortly thereafter brought some really terrifying news. A Florida man, Robert Stevens, had died from inhalation anthrax, a very dangerous infection of the lungs that spreads very quickly throughout the body. He was the first U.S. anthrax case since 1975, and the only of one of 18 cases in the 20th century. Now, everyone on the committee, this biological threats committee at the CIA, knew a lot about Bacillus anthracis. We refer to it as anthrax. And our first reaction, quite normally, was that this was a rogue nation that was at work, because this was right after the 9/11 incident of the Twin Towers. And we figured that it was either a homegrown terrorist group or some crazy individual. But given the timing, so soon after 9/11, the committee, I and the others on the committee, and many in the government just assumed it was Al Qaeda. And it was just a follow-up on the World Trade Center and the Pentagon attacks. And of course, there was some logic to our thinking. Well, when the president was told about the Stevens illness, he assumed it was part of a major biological weapons attack. The government health officials moved quickly to assure the nation that there wasn't really any need for concern. And Secretary of Health and Human Services, HHS, Tommy Thompson, repeated 6 times on national television that Stevens' illness was an isolated case. And he even suggested that maybe Stevens might have gotten sick from drinking stream water during a hike in North Carolina. That was unfortunate because it made the government look really uninformed, rather clueless, really. Anyway, I won't go into details about anthrax. It affects really primarily large grazing animals, cattle, sheep, goats, pigs, elephants, and in northern Canada, even bison. It spends most of its existence in soil, usually in a dormant state. You can think of it sort of as hibernation. And when an animal grazes on the grass, it can breathe the spores in its nose and get infected. Anyway, I was the first microbiologist to direct the National Science Foundation. It was very fortuitous because I had a very unique opportunity to bring my scientific expertise to this terrible incident. Now, the NSF funds research, but it doesn't really do research at the foundation building itself. And so, but we know all of the researchers around the country. So I very quickly offered John Phillips and the intelligence community the expertise of the NSF. And John and I created a program to use the CIA's deep pockets to help finance publicly, and with no strings attached, scientifically worthy projects that the NSF couldn't afford to. That is, by the time it had run out of the funding, there was still some really good projects. And if they could be of interest and help to the CIA, then funds could be transferred. And we made this arrangement. In fact, some of the first grants were not of the anthrax, but from NSF, from this program, were for Larry Page, who went on to found Google, and Sergey Brin in 1998. When they were still graduate students working on a search engine. Anyway, what we did was that time passed, and I'll make this, I won't go into too many details here, but I knew that we needed to sequence this bacterium. Now, this is at a time when we just had a few sequences. Craig Venter had cavalierly sequenced the genomes of 2 bacteria, Mycoplasma genitalium and Haemophilus influenzae. And he was thinking about sequencing the very big genome of Bacillus anthracis, and they had just started but hadn't really gotten through it. To make a long story short, in this particular aspect of the study, we decided that we learned that unfortunately the anthrax that had killed Stevens was in fact a strain that had come, it was called a particular strain that had come from the US. And with studies that we did, we were able to determine, it was able to be determined that this particular bacterium was really a homegrown strain. So after that initial admission of Stevens to the hospital on October 2nd, on October 12th, 10 days later, the situation really changed because then Tom Daschle, Tom Brokaw, the NBC news anchor, his office received a powder in a crazy misspelled letter. This is next, take penicillin now, death to America, death to Israel, Allah is great. 3 days later, it was when Tom Daschle, his office, his secretary opened a letter with a powder, but she was by this time alerted and held the envelope at arm's length, and that quick action saved enough powder to be tested. And then more anthrax letters appeared, and it was pretty clear that we now had a really dangerous situation. I called late in the evening. I called Claire Fraser, who had taken over as president of the company that Craig Venter had formed, Tiger, the Institute for Genomic Research. And I was trying to be circumspect to say, look, you know, if somebody should submit a proposal to sequence Bacillus anthracis, it would be received and reviewed very quickly, because at that time we had a program that we could provide funding very quickly within 2 weeks with internal review for startup funds. But apparently Claire didn't quite get the point. But after the series of incidences, she then called and asked if funding would be available. Of course, we immediately made funds available on October 26 in 2001. And our research was begun with Claire Fraser and Timothy Reed at Tiger. By this time, we knew that this was an Ames strain. It had not been developed by Al-Qaeda. It was a homegrown strain. And subsequently, in the various studies at Fort Detrick and elsewhere, a very bright technician learned from her studies that her name was Teresa Abshire. And Terry Abshire was very knowledgeable and realized that there were some mutations in the colonies that appeared on the growth medium that she used in the laboratory at USAMRID out at Fort Detrick. And as a result of the work that she had done, we were able then to at least understand that sequencing would be important. Now, the time was going on, and there wasn't anything happening in the government. And finally, on December 18th, Ari Petrinos, a leader in the Department of Energy, where the Human Genome Project had actually started, I had called him, and I'd also called Tony Fauci and said, look, we've really got to sequence this, because my background as a microbiologist, I had written a computer program in 1960 for my PhD thesis to analyze and identify marine bacteria, including Vibrio cholerae. So I knew that we really had to work fast and get moving. So Ari took it on himself to get a group together in Tony Fauci's office. We all met, and then during that first meeting, we reached a quick consensus. The country needed a database of genetic information on pathogenic microorganisms. Petrinos was very interesting. He was very pleased with the meeting, and he often kept two sets of notes, one in English that snoopers could read over his shoulder, and the other a secret diary his native Greek for his eyes only. And so after the meeting, Petrinos wrote in Greek that he was very happy with this outcome of this meeting. But then nothing happened. I waited another week and I called Tony and said, look, I'll run this thing. He said, fine. He said, go for it. And so he delegated Maria Giovanni, a very talented genomic scientist at NIAID, as his liaison. And Jack Marburger, who was the science advisor, someone whom I had known well before he had become science advisor, he assigned Rachel Levinson as the liaison with the White House. And so I ended up leading an interagency team of experts who had power of the purse and hands-on knowledge. And I realized very quickly, this could not be a normal committee, because if we were official, and we could have had a presidential directive, we could have had easily any kind of formality of organization. But then if we were an official committee, we'd be subject to FOIA, the Freedom of Information Act, and the FBI and the CIA wouldn't attend because they were addressing a criminal act, and they could not have the minutes of the discussion made public. So at our very first session in early in 2002, we agreed that we would keep no minutes, no public hearings. We were in a kind of bureaucratic limbo. And we met every week for 3 years. It was every Friday afternoon in a skiff in one of the second building at the NSF. That happened to be a company that had a skiff that we were able to utilize. And we met every Friday afternoon from 1 to 2, because if we were later, the representatives from the FBI laboratory in Virginia wouldn't get home till 7 or 8 at night if the meeting ran over. So we had very tight meetings every single week, Friday afternoon. We called that our research group meeting, and we called ourselves the National Interagency Genome Sciences Coordinating Committee. And so with representatives from every agency, NIH, DOD, EPA, USDA, NSF, it was about 15 or 17 representatives, and we met, and our first priority, of course, was anthrax. But what we really did was to build up a complete library of sequences of all of the known pathogens that could be bioterrorist agents. We created the list. We simply said A were those pathogens of severe danger like anthrax, smallpox, etc. And then B were the ones that could create serious problems like adding salmonella or shigella to, let's say, a salad bar in a supermarket. You could really create a lot of problems. And then thirdly, C group were the animal and plant pathogens, because of course, if anthrax was spread around the herds of cattle in Texas, we'd have a catastrophic failure there. In any case, I won't go into all the details, and I'll close for discussion. But because we had this team of experts working together, for example, Bruce Bedoli, the FBI's leading DNA expert, and someone I think who did the most to modernize the FBI's approach to science, he took time off from identifying victims of 9/11 to help on this study. And so while we were doing our detective work, we ended up over the next several years, actually, 2 or 3 years, developing the very elegant sequence of the anthrax strain plus other anthrax Ames strains. It was called Ames simply because when a heifer some years ago had come down with anthrax, and it was in Texas, but the envelope that arrived at USAMRIID had Ames written on it, so it got called the Ames strain of anthrax. In any case, investigative work was done, and the sequencing was completed. It turned out that Jacques Ravel, a former student of mine, who joined when he finished his PhD, joined Tiger. He ended up taking over from Timothy Reed, who went on to a position in Georgia, and the work that was done by Jacques Ravel and his team was to determine a set of about 8 mutations that had a very characteristic distribution. And by matching it up, and the team at Tiger, Mihai Pop and Adam Philippi, a number of people at Tiger working along with Jacques and his group. Jacques remembers very well in September 2007, this was 6 years after the anthrax attack, he reported the latest results to the several FBI agents when one of them asked, what about sample X? Can you show the results there? Now, Ravel knew that sample had already tested positive for 3 mutations. And just that month, he found that it had tested positive for 4 of the key mutations. And when the agent saw the result, Ravel watched his expression change from neutral to extraordinarily happy. And Jacques said, I could see that particular sample meant something. Now, poor Jacques is a Frenchman from birth, an American by citizenship. Normally, he would have celebrated with his lab with champagne, but he couldn't tell anybody. He couldn't tell anybody that the agent had smiled when he reported those results. Anyway, we finally got the vital evidence. 4 genetic changes had been identified as the fingerprint of the mailed spores. And 8 of the 1,070 Ames samples collected by the FBI had all 4 of those changes. With that knowledge, the FBI then used the traditional detective work, and by mid-2007, the FBI clearly could say the only Ames samples testing positive for the 4 genetic changes came from one particular flask at USAMRIID. And Worsham and Abshire, the 2 women, had identified the first mutations. And the flask was secured by magnetic card access and stored in an anthrax vaccine expert Bruce Ivins' walk-in refrigerator. By mid-2008, the FBI had established that Ivins was the only person who had ever worked alone around anthrax samples from that flask. Unfortunately, we'll never know for certain what exactly happened and whether Ivins was working solo or with others, or whether he was really truly involved. Because as the FBI was preparing to arrest him, he took enough Tylenol to put himself in a coma, and he died 3 days later on July 29, 2008. On August 18, 2008, the FBI had a press conference, and then 2 years after Ivins' death, the FBI published the 92-page Amerithrax investigation summary. So that was one chapter amongst the many adventures I've had in my life, but it's one that I thought you would be interested in learning about. So I will pause there and be very happy to discuss with my colleagues.
Rita, I don't see, oh, there's Yona.
Yeah, well, that's okay. I wonder, Professor Caldwell, can we invite Norm Kahn at this point, perhaps to become involved in the discussion
Yes, please.
related to that because it's quite relevant. Norm?
Okay, Yona, thank you very much. I am absolutely honored to participate in this celebration of the publishing of Rita's sterling memoir. During the course of my career, I had the pleasure of working with Rita on numerous occasions, and I learned much and I benefited greatly from her experience and expertise. So Rita, I'd like to follow up on your national security related comments that you just gave by posing a few questions to you on the intersection of bioscience and national security. And I'd like to start off with a two-part question. What are your thoughts regarding the national security implications of the recent advances in genetic engineering, such as CRISPR? Specifically, what threats can you foresee in terms of misuse by states and also by malicious groups or individuals? And then part two, what do you think we might do to mitigate the threat? So over to you.
Very good. That's a good question because there is a concern about bioengineered organisms, but you know, it's really interesting. I think Mother Nature does a much better job in producing biothreats. And I think really to the second part of your question, I think there's not a whole lot we can do, but we need to look for, let's say, anomalous kinds of activities where there might be insider threats, being aware perhaps in a large pharmaceutical company of behavior or comments or whatever of an individual that might suggest that there might be something afoot. And in the case of the lone stranger, so to speak, I think it's very hard to be able to identify such people. However, the possibility that an individual can really work on his or her own on genetically engineering a bioterrorist weapon, I think is pretty slim. Because when you consider the work that goes into CRISPR, for example, which would be a tool that would be used if you would think of engineering an organism, that takes a lot of sophistication and a lot of elegant kind of laboratory apparatus and manipulations. However, it's not so difficult to grow anthrax. And so I think it wouldn't be so much the bioengineered version, but more likely looking out for someone who has the ability to grow in his kitchen a bacterium. And then there, I think we just have to look for anomalous situations, anomalous purchases or whatever. Do you think that would be what you would interpret, Norm?
Could you say that again, please, Rita?
Would that be the answer that you would expect?
Yeah, generally speaking, yes. I think your comment that there's a fair amount of sophistication required to do genetic engineering, whether it's CRISPR or otherwise. I think that's an excellent point. My concern would be an individual like a Bruce Ivins, someone who actually has the appropriate skill set, to do that kind of relatively sophisticated work. I think I look at the intersection of intent and capability. So if your intent is really bad, but your capability is low, nothing's going to happen. Which I think is to your point. My concern is on the high end, where someone like a Bruce Ivins, let's say, imagine not a Bruce Ivins, but a competent virologist, who for whatever reason goes off the reservation, and at that point you have a problem.
I totally agree. And there, I think, is where you have to look for anomalous behaviors.
Yes, exactly. Yes. As a follow-up on that, before I get to my next question, in terms of minimizing or mitigating this kind of threat, where do you place the importance of ethical training of scientists?
I think that's absolutely key. That's absolutely key because several organizations, the National Academy of Science, the National Sigma Xi Scientific Honorary, and just about all of the scientific and engineering societies have developed a code of ethics to be a scientist. What is expected and what actions and activities are unethical. For example, to engineer, let's say, a toxin into E. coli just for the fun of it, that's not a particularly ethical activity. And therefore, I think ethics training is really necessary these days. Well, it always has been, but even more so now for young scientists.
Couldn't agree more. Okay, next question. This addresses the potential of behavioral modifications through biochemistry. What are your thoughts on the risks that such modification may pose in terms of societal influence or disruption? Is that something that ought to be a concern?
I really do think that is probably a 21st century danger, because I think we now know that just from the studies that I've been doing lately with the microbiome, it's amazing how the influence of the gut and the microorganisms of our gut is now considered almost another organ of the body, can influence the brain. And these compounds are the ones that make you feel good or feel bad or feel ill or whatever. And I think as it becomes known that you could produce a spray of certain kinds of chemicals, you could even modify the behavior of a crowd. I think, and that sounds a little, super scientific-y, but it's not out of the realm of the possible. And I think just moved control and understanding the social behavioral sciences. I think we have underinvested in the research on the social behavioral and economic sciences that drive human behavior. And we really need to understand this well, not to control necessarily, but to understand and to provide it as part of wellness of individuals and of populations.
Yeah, this is Vince Cerf. I just can't resist breaking in for one moment and saying that this last bit of discussion provides a whole new definition for "I go with my gut."
Yes, it does. It does indeed, Vince. Welcome to the discussion.
Thank you. Sorry to be late. I was on a UN call that I had to couldn't escape from.
Do you have any questions?
Okay. Rita, I've got one more question for
Sure.
you, if I may.
Yes.
This is a much broader question, and it brings into play the whole idea of the bioeconomy as it relates to national security. And what I'm thinking about here is the reality that a strong national security posture is predicated, among other things, on a strong economy. And so how do you think we stack up to our competitors when it comes to the development of our bioeconomy and the investments we're making?
I really feel strongly that we need to learn from the shortcomings of developing nations where there's a chasm, a very wide chasm between the ultra rich and the very poor. This creates a dissonance in society, and it needs to be addressed in a way that certainly fits within the ethics of the economy as we like to consider it. But I feel very strongly that again, as I said, understanding economics and the capability, the technical capability. This is where I feel strongly and have written a book about sexism and misogyny, because you don't fight a war with half the army. You don't fight a technological competition with half the brains. And it's pretty clear that the Y chromosome doesn't carry intelligence genes. And so if we're going to really mobilize the entire workforce, regardless of sex or gender or race or whatever, we really need all the brains. And so I feel very, very strongly. And that's why the book ended up with the secondary title of A Journey Through Sexism in Science, because we have to overcome these inhibitions to bring women, but also the underrepresented, into the bioeconomy or the economy in general. And we need to balance things. I think is critical.
Great comment. Thank you. Yona, I'll turn this back to you.
Okay, you're welcome to join us on other issues. May I again formally invite Dr. Winton Cerf to join us, and we try to introduce you briefly. Certainly, it's very well known, widely known, that you contributed as one of the fathers of the internet and so forth, and your very exceptional work in the field. Could you kindly join us and see if you want to follow up with Professor Caldwell at this point?
Yes, I'd be happy to, and I thank you very much for the opportunity to do so. Rita, it's always a pleasure to have any time at all with you, no matter what the circumstances. And I had a lovely time reading your book. But I have to admit that, first of all, I had not understood that there was an angry Colwell hiding behind the facade that I got to know at Cosmos ID and at NSF. Wow, talk about powerful energies. But more important, I think, the compelling stories that you told about your experiences as a woman and others who have experienced this terrible, it's worse than bias. I mean, it is just totally unfair treatment and wrong treatment, was very compelling. And so I'm glad that you felt comfortable sharing some of those painful stories with the rest of us to simply reinforce the importance of correcting them. I did have a couple of questions, though, particularly with regard to the cabal that you set up in the Friday afternoon skiff. First of all, I absolutely agree with the tactic that doing something informal was actually important. There's a parallel example of this in my world. There's something called the Configure worm, which is essentially a piece of malware. And it popped up quite some time ago, and a very informal group got together to try to figure out what it was and what it could do, and maybe even where it came from. And the group was deliberately set up to involve, government people and private sector and so on, with absolutely no formality whatsoever, because we all assumed that as soon as you introduce requirements for participation and everything, bureaucracy would suddenly set in. And so informality turned out to be incredibly important to draw hackers into the story and people from outside the US and so on. So I think your intuition was exactly right there. Do I remember correctly that Josh Lederberg might have been part of that discussion? Because I remember that he was very worried about the abuse of biotechnology in a variety of different ways.
Josh was not, but several of his, not exactly students, but his colleagues who had worked with him were members of it. But he himself, because this was a government agency group, these were representatives from all of the government agencies. And what we did, what I didn't point out, unsure, is that literally we just pooled our money. That is, I just had an easel and I just put A, anthrax, who can fund it? And then each put their hand up, Maria, Giovanni. And then when it came to each one of these, Homeland Security would pay for some, and the NSF paid. So that was how we pooled the money. It was very informal, but these were people, not the heads of agency, because they wouldn't know anything about.
Exactly. Too much visibility, right? Yes.
But these were high enough that they had budgetary control.
Yeah, this is not unlike the small consortium of NSF, Department of Energy, NASA, and DARPA, who pooled their resources in order to enhance the growth of the internet, which NSF, of course, had a great deal to do with.
Exactly.
So there was the reason I mentioned Josh, though, is that I remember having a conversation with him about a genetic modification that he speculated about, which was to somehow infect people in such a way that their bodies would generate narcotics and leave them in a state of utter euphoria. Of course, if you do that to the military, you might easily overcome any kind of opposition, because the opposition would be too busy off in cloud la la land. And I remember thinking how terrifying it was to imagine that might even be feasible. Is it in fact possible to do that?
I think it will eventually, and that actually was part of the question that Noam Khan asked. Is it possible to behavior modify? And that is one of the mechanisms by which to do it. We now know already that there are compounds that are produced by bacteria in the gut that pass the blood-brain barrier. It's already known. And they influence the sense of well-being that you get from the nerve cells in the brain. So yeah, I think we need to be aware of this and understand that the breakfast cereal that may be distributed might have maybe a certain lactobacillus species that has been engineered to have a capability to produce what's that you get when you go out running? The good sense.
Endorphins.
Yes, the endomorphins. So there you are. There you are.
Wow. So I could just imagine the cereals containing psilocybin and peyote or something, which we've already discovered do these interesting things, and we've known that for presumably centuries, if not thousands of years. Wow. Well, this is all very scary. Let me come back actually to not to the national security topic, but to the one that you spent a substantial amount of time on in the book. Are you seeing significant shifting now in this gender bias space, or are we still, the statistics still do not look very attractive, although in the bio space, it looks like there are more women who are successfully being involved than in, for example, computer science and engineering.
True. The computer science and engineering have improved a bit, or at least physics. Physics now is in engineering about 17% of population, which is trivial. I mean, we're losing a lot. I think it's changed in that when I was an undergraduate and I was an A student, I'd been accepted to medical school, to 3 actually, Yale being one of them. And I was on my way, but I happened to meet this handsome graduate student my senior spring semester of my senior year. And Jack and I decided to be married. We were married before I graduated, and we were married for 62 very happy years. I still miss him.
Wow.
Anyway, we decided that it would be better for him not to lose a year of graduate study. And I really didn't want to, I mean, I love Purdue, it's a great place, but I didn't want to do my bachelor, master, PhD at the same institution. It's a good idea to move on. So I went to my department chairman and said, could I get a fellowship? I'll do a master's and Jack will do a master's and then we'll move on. And his remark was, we don't waste fellowships on women.
Jeez. Several bad words occur to me right now.
So I went to my undergraduate advisor who knew I was a good student, and his reaction was, oh, well, he said, their loss is our gain. He said, I've got an RA, research assistantship. If you'll take care of the fruit flies, you can do a master's in genetics. So that's how he ended up going into genetics. So I don't think anybody will be that blatant. They might say, well, we don't have any left available right now or whatever, but they won't be so crude. So but now I think I classify these kinds of things as going from the criminal to the clueless. The criminal you can recognize. I mean, physical attack. I mean, that belongs in the courts with police. But the clueless are the things that wear you down. It's when you're overlooked, or when you say something at a meeting and nobody says anything about it, and then a guy says the same thing you just said, and everybody says, what a wonderful idea. These kinds of things can be very depressing. So I think that hasn't gone away. And what I recommend when I speak to women, which I do now, invited to lecture, I say, look, get a posse, get a group of friends, and talk over your experiences. You'll find out that the experiences you thought were your own are shared by almost all women. And this is a good example. Nancy Hopkins at MIT 30 years ago.
Yes.
The famous study where she just went around and measured labs, found out that men had almost twice as much space to work in than women did.
So didn't Charlie Vest respond fairly well to that? Am I remembering correctly?
Chuck Vest was super. He said, he felt that, I'm paraphrasing, that there was some substance, but he didn't realize how much and how significant, because Nancy had gone to him with 15 women faculty at MIT. There was only 16, and one woman felt that she didn't have any problems, but the other 15 agreed, and they all went together. And Chuck was great. He made changes.
So get your data together and form a posse.
There you are.
Any other question? No?
There are some questions in the Q&A system if you happen to have noticed that.
Yeah.
Rita, this is Robert Sargent. We do have two questions if we have a moment.
Sure.
Before we move on. First question is, were there any anomalous behaviors observed in the anthrax case?
There were really many anomalous behaviors of Bruce Ivins, but they didn't reach the point of recognition. Things have changed since then. I was asked by the federal government to testify because the widow of Robert Stevens sued the government. And I got to read the full psychiatry report. And turned out Bruce Ivins was very, very strange. He did a variety of things that were not particularly normal. He, for some reason, had an idée fixe about Kappa Kappa Gamma sorority, which is kind of strange. And it's written up, I think, in that part of it, his behavior is written up in a couple of books about the anthrax story. He also, during the last, before he committed suicide, he had been required to see a psychiatrist, and he had threatened to take out his enemies. So we were lucky that we didn't have a major incident before he was apprehended, or at least, unfortunately, committed suicide. So there were some behaviors, but they were considered to be just kind of ascribed to a scientist who spent all his time focusing on his science. Nowadays, I think we recognize that there are some behaviors that we which is what Norm and I were referring to, anomaly, where you really want to go to an ombudsman and make a report and let there be quiet without a public, charging of any activity which would then affect the reputation if it were perfectly strange but not abnormal. So I think we are wiser today than we were 20 years ago.
We do have one additional question that's multi-part. I'll be happy to repeat it as necessary. First of all, a comment, a great discussion so far, very informative. The questions are, any thoughts about the ongoing question of the origins of COVID-19, natural or lab grown? Second issue is, to follow up, what are the dangers of similar viruses being used as bioweapons by terrorists or state actors? The next issue is, what agencies in the US government are tasked with tracking and assessing that threat?
Well, first of all, I know that there's a script that is sort of floating around about someone in the Wuhan laboratory having genetically engineered the virus. We now have, at first, it was the sequence that was provided by the Chinese laboratory, Chinese government from one of their laboratories in January. But we now have at least 240, probably 300 sequences of virus now, sequences that have allowed us to see variants and so forth. And there is no evidence of genetically engineering work being done on the virus. We now have the best engineer of all, Mother Nature. And we now know that there are variants, and there have been some mutations on the protein on one of the spikes that apparently makes it more transmissible. So I think that the likelihood of this particular virus having been engineered and released either inadvertently or advertently is in my view, zero. I don't think that is the case. And again, I think that, as I said in the discussion with Norm, we both agree that right now, for someone to, let's say a rogue or a loner, to engineer effectively and release is highly unlikely. It's the insider who is highly trained. And there again is where we have to be aware of anomalous behavior or threats or what is done for clearances. We probably need to do this for individuals who work with highly pathogenic organisms. That is, make sure that they don't have any behavioral problems such as excessive debt or bad home situations that would make them behave abnormally and carry out some kind of threat. So the answer to the question is that I think it's very unlikely because the group that has been working with Wuhan, the EcoHealth Alliance, has been working with them for about 20 years. And as a result of that collaboration, we now have remdesivir. That's a result of their work collaboratively. And the evidence is that bats carry a variety of coronaviruses. And so it's more likely that the biggest engineer of all, Mother Nature, will have another surprise for us. Maybe it'll be COVID-20, COVID-21, or whatever. So I think because we are moving into unexplored areas, we're breaking down in areas that humans have not lived before, and we're exposing ourselves to pathogens that we as humans have not been exposed to, and we're going to have some surprises. And I think also we're destroying biodiversity, and biodiversity is not a hobby. It's not something that we, is just pretty to look at. Biodiversity and the chain of connections keeps us healthy as humans. And so the loss of biodiversity is a threat to our health. And we need to understand that all of life on the planet is a system, and it's highly interconnected. And just because we lose a snail darter, we shouldn't just be amused. We should be worried, because there is a connection between it and other levels of life. And so I think we need to be paying attention really seriously to climate change and what it's doing to our climate, but also to diversity of life and the selection that it's carrying out while we blithely go on with our daily life and not paying attention to significant changes. For example, warming of the oceans, which lead to loss of some of the protective components in the oceans. I could go on and on. Have I answered all three? I tried to keep on.
Yes, the last question was, what agencies in the U.S. government are tasked with tracking and assessing this threat?
Well, I'm going to assume that the intelligence agencies are continuing to monitor threat. If they're not, they should be. And this includes the defense intelligence as well as the CIA and the various other of the 17 components of the national intelligence. And I think it's really, really critical that this be done, especially during the current disturbing times when the nation seems to be so divided. I think this opens opportunity to those who would like to do us harm. And we really do need to pay attention to the potential of release of biothreat agents. And we really should be having some group working as we did have 20 years ago. I'm assuming that it does continue.
Rita, may I ask you a follow-up question to the previous one that you mentioned, the COVID-20 or 21, what's next? The question is whether the prediction model that you try to chart in regard to the cholera work could be applied somehow to the next threat of the COVID, whatever is going to develop.
Well, we've successfully adapted, I should say, the model that we developed for predicting cholera, which, by the way, we are using in collaboration with the British Aid Agency and UNICEF to provide a 4-week advance on the risk of cholera in Yemen. We were able to show where the risk is highest, and we provide them with a monthly, I guess every 2 weeks, we provide them with a map of the risk for 4 weeks into the future, so they can be prepared to locate medical supplies, physicians, and safe water in those areas. And we've seen cholera reduce significantly in that ravaged country that's under strife with civil war and all kinds of problems. And what we've done is to take that satellite sensor-based predictive model, and we now can predict in counties of the US risk of COVID-19. And I think it would be interesting to do what you're suggesting, Yona, and that is to predict what sort of threat agent might, with this particular pattern of behavior as the COVID-19 would emerge in another year or two. It would be an interesting exercise to carry out. So maybe I should go to the funding agencies and suggest it.
You mentioned, I think, that you worked also with some of the UN agency, for example, related to children, UNICEF and all that. Could you elaborate a little bit on that?
Yes, this turned out to be very interesting. What happened is that we had been developing this model. My first predictive model was 20 years ago because I had determined the relationship of the cholera bacterium to copepods. And the copepods graze on phytoplankton, which carry chlorophyll. And Landsat provided sensors that measured chlorophyll, sea surface temperature, and sea surface height. So 20 years ago, I was able to correlate the cases of cholera in Bangladesh with these changes measured by satellite just off the coast in the Bay of Bengal. And so we have perfected the model to the point where we did a retrospective analysis of cholera in Haiti, and then did another cholera in Haiti when Hurricane Matthew went through Haiti and up the east coast of the US. And we did a retrospective analysis, and the fit was so good, we published it. And it was picked up by Scientific American, and a colleague in England, a Scotsman, read the Scientific American squib, called us up in January of 2018 and said, hey, could we work together? So that's how we started, by providing them with a predictive map of risk. And then they located that this was the British Aid Agency and the British Meteorological Agency, and was funded by NASA. And we connected with UNICEF. And so, working collaboratively, we are able to send out these alerts that allow then positioning physicians and supplies. And this continues today. So in fact, I just received today the latest printout from the collaborative team, Dr. Anta Judla and his students at the University of Florida, Gainesville. He's an associate professor tenured in the School of Engineering. He's an engineer, and he was a postdoc at Maryland, and then we've continued to work together.
So you see more collaboration, cooperation in the science, technology around the world?
I think we have to do this for public health. Public health is global, and public health has to be international, and it has to be interdisciplinary. And I think that one of the things that we're adding to the satellite study is we're monitoring the sewage for COVID-19, SARS-CoV-2 virus. And so that gives us ground truth. And this is really makes it very elegant as a very powerful public health tool. And I think that one of the benefits, if there are any, of COVID-19 is that it has, I think, put attention to what should be added to the kinds of measurements that we do when we go for a physical. We give a blood sample, we give a urine sample. Now we might want to be giving a fecal sample. We do, in effect, in a way, because until colonoscopies became so common, we used to do a kind of a test for colon cancer that involved a stool sample. But we should consider the stool sample as another part of understanding the well-being of humans. And I think in the future, this will be common and normal.
Rita, this is Robert Sargent. I apologize. We have one additional question, and this will go to not only yourself, but the other panelists who might have interest in answering. The question is, what would the panel identify as the top 3 bioterrorist threats today and in the near future?
I would say that there's always the bio threat of a pathogen, but I think it's becoming much more sophisticated. And I do think the bio threat of behavior modification is real, and it needs to be studied. And then I do think I consider it a threat to national security, social stability, and economic strength, if we don't address the disparity in our economy between the haves and the have-nots. We simply must, and that's a threat. But Norm and Vint, I'd love to hear your comments.
Well, I echo your thoughts, Rita. This is Norm. I certainly echo your thoughts. I think it's very difficult to have a top 3 or a top 10. And I would get back to something I mentioned earlier. It really, in terms of the impacts such an event would have, what you look at is the balance between intent and capability. And so if someone has very, very high capability, they could do very, very bad things. And if someone does not have that capability, if they don't have the training or access or understanding of what we call tacit knowledge, how to really make something work and how to troubleshoot it, they may have terrible intent but won't be able to pull it off. And so I think there's this huge range of possibilities, and I really can't begin to think on how I would rank them other than just intent and capability. But again, echoing your comment, Rita, the idea of behavior modification, which gets away from the traditional pathogen-based threat. I think it's really important to understand that biothreats are not necessarily directly pathogen-based.
So it's Vint. I'm just thinking a little bit about the problem of detection. I'll stick on the pathogen side for just a second, even though Norm's point about modification is pretty scary. But one thing that we might have been able to detect the presence of this particular COVID-19 disease sooner if we had been analyzing water samples, for example, in the sewage processing systems. And so it does suggest to me that in introducing analytical capability, especially in our metropolitan areas, might actually be a useful and cost-effective thing to do.
I would agree with that strongly.
I'm sorry, as long as I got this microphone here, could I just ask another question of Rita and the other panelists? And this has to do with the global warming and the side effect of the permafrost, thawing of the permafrost and the release of methane, but also possibly ancient pathogens that haven't been exposed to, or we have not been exposed to for a long time. How serious is that problem? I know there's been at least one report of anthrax release in Siberia, but I don't know whether this potential is simply a scary story or whether we should actually be conscious of that and do something about it in terms at least of detection.
My reaction is that it's a definite threat. We've already recovered the 1918 influenza virus. That was done by a corpse having been exposed with the melting of the snow. And then the virus actually isolated and sequenced and identified. What I would really worry about is, for example, an old pathogen that was terrible, and that's smallpox.
Oh my, yeah, which we thought we got rid of, right?
That's right. And we're all, well, us oldies were vaccinated. We'd have residual immunity, but just think of the entire globe would be totally susceptible to smallpox.
Wow.
So that's a nightmare for you.
I wonder whether the consequence of that logic suggests that eliminating a toxin or a pathogen like that is actually potentially hazardous, as opposed to having a more general built-up resistance to it.
I'm one of those who feel that it is a hazard. I'm one of those who feel that we should have a stockpile of the vaccine. Norm, what do you think?
Yeah, I concur. Yeah. I mean, if you do eliminate something, you need to keep the capability to deal with it should it resurface.
So what would that imply in terms of stockpile? Because the most recent reports of the vaccines for COVID-19 are that they have to be kept at minus 100 degrees. And that's a fairly intensive and expensive proposition. I wonder whether there's anything to be done to find vaccines that are storable at room temperature.
Well, I think that it would be definitely doable, particularly for smallpox, because the vaccine was pretty crude. It was a killer virus. I mean, Jenner simply took cowpox from a cow, and that was pretty crude. So I think it is possible, and that's why it's a pity that it's not being done.
I think, sorry. The Moderna vaccine is less constrained in terms of temperature than the Pfizer vaccine. But also, I think what we're seeing now, these are just first generation vaccines. And to your point, Rita, I mean, I would expect follow-on vaccines, to be more amenable to less rigorous constraints in terms of storage.
Yeah, the second vaccine that's just been announced as being 95% effective does not have the severe cold chain.
Right.
Oh, that's good to know. Now, some people are suggesting small molecule treatment in order to interfere with reproduction or ACE2 connectivity. But I wonder what's the implication of trying to use that kind of a small molecule treatment. It doesn't reproduce itself, unlike the antibody effects of normal vaccines. So does that just imply lots and lots of shots over a period of time in order to maintain a level of small molecule defense against the virus?
Well, I think it will, because similar to the flu, we get a flu shot every year, though there is an attempt to get a multivalent influenza vaccine. But what I worry about with the new first generation type vaccines that are coming out, where we're dealing with RNA molecules, I just wonder whether we'll have any surprises. It's sort of like the using the gene splitting technique and producing a child in China, which I think the scientist was rightfully jailed, because we don't know what other side effects. I mean, you could be affecting the regulation gene and not know it, of some other metabolic process. So I think we need to proceed very carefully. I see Noam nodding. You agree?
Yeah, I agree. I mean, the one thing we know about biology is that whenever you think you've got it figured out, there's something else that pops up and you realize you didn't quite get it the way you thought you did. So.
This, Norm, this is theorem number 206, which reads, everything is more complicated.
That's right.
This is Robert Sargent again. I do have one more question that has just come up. This one has a little bit of a technology bent to it. What is the intersection between the internet and bioterrorism? Thinking about behavior modification or bad actors sharing information on how to produce and distribute pathogens and toxins.
I think that is really fascinating, because there you're inducing the endomorphins or endorphins or whatever, not by injecting anything, but by transmitting to make a group feel good or behave as a mob, as we all know happens. This is behavior modification without the bioinjection. So I think there are people. In fact, Vint, this is your turf. You probably should be answering this.
Well, considering that the internet got mentioned here, I feel compelled to at least say something. I will tell you that there are a lot of people who say, look what I found on the internet, this is horrible. And then I have to say, by the way, have you been to your public library recently? So, it's very hard to hide knowledge once it has been discovered. And so I don't mean to make fun of the point, which is that there are hazards associated with learning how to do X, Y, and Z. But I'm not sure that trying to suppress information is the successful defense. I think we just have to do a better job of indications and warning, intelligence gathering, and good old-fashioned law enforcement.
Actually, when we had sequenced the anthrax, my first reaction was that hadn't we better hold this tightly for a while anyway? And interestingly, John Phillips, and I think you too, Norm, said, no, this has to go out, because only the bad guys will get it anyway, and we want to make sure that the good guys have access to the information.
That's a good point.
Mm-hmm. Yeah.
Remember that, Norm?
Yes, very much so. Yeah. Mm-hmm. Yeah.
Professor Don Wallace, would you?
I'll say something. Well, Yona, listen, thank you once again. This has been a really wonderful and searching discussion. C.P. Snow, the writer, once said, we have the illiterate and we have the innumerate. And it occurs to me we also have the scienceless. And I think one thing I've learned from this is that, we've been very worried about sort of the limited knowledge of our public. And when you think of climate change, not to mention the pandemic, I think it becomes absolutely essential that we educate ourselves a great deal better. And Rita, I've listened to you now for several years. I've never thought of you as a woman as opposed to a man. I've thought of you as having a superior brain. And now I've learned that you've applied it. Anyway, Yona, have more of these wonderful programs. Have Rita back. And thank you all very much.